Welcome to Dr. Balga's podcast for the Curious Doc. Today is 25th February 2026. This video is on a Nature article, open access, published online today, titled Human Hippocampal Neurogenesis in Adulthood, Aging and Alzheimer's Disease. There's an accompanying associated news article titled Brains of SuperAgers are still strong, producers of new neurons. The study was human hippocampal neurogenesis in adulthood, aging and Alzheimer's disease. Young adults, 8 individuals, 20 to 40 years. Healthy agers, 8 individuals. Preclinical Alzheimer's, intermediate, 6 individuals. Alzheimer's disease, 10 individuals. And superagers, 6 individuals comprised the study cohort. The total nuclei analyzed were 355,997 from post-mortem hippocampi. Techniques used included single-cell RNA-seq and single-nucleus assay for transposase-accessible chromatin sequencing, analysis of differentially expressed genes, differentially accessible regions, and enhancer regulon mapping. The statistics used included Benjamini-Hochberg corrected false discovery rate for p-value for value less than 0.05. Mann-Whitney U-test and Q values were reported. What did they find? Neural stem cells increased in preclinical and Alzheimer's disease cohorts. Neuroblasts and immature neurons significantly reduced in Alzheimer's disease versus healthy agers. SuperAgers showed a 2.5-fold increase in immature neurons. Neuroblasts increased versus Alzheimer's disease Q value 0.0002. Most diagnosis-driven alterations reflected chromatin accessibility rather than gene expression. Early downregulation of synaptic plasticity DARS observed in preclinical cohort. Limitations of study: postmortem design, cross-sectional, variability in human samples, causality cannot be inferred. What did the news article say? It reported that neurogenesis persists in adulthood. New neurons comprised of about 0.01% of hippocampal neurons. Alzheimer's disease brain showed fewer immature neurons. Superagers had higher numbers of immature neurons than other groups, and significantly more than Alzheimer's disease. The cohort size was small, less than 10 per group, therefore limits the statistical power. Epigenetic profiling using single-nucleus RNA-seq and chromatin accessibility assays strengthened the specificity. The implication: enhancing adult neurogenesis may represent a therapeutic target for cognitive decline. Clinical pearls: chromatin accessibility changes precede overt transcriptional decline. Synaptic plasticity pathways represent early therapeutic targets. Neurogenesis persists into adulthood in humans. Superager molecular signatures suggest resilience is biologically encoded. Epigenetic modulation may represent preventive strategy. These are some eponyms and concepts related to this paper. Podcast brought to you by masterbedfacts.com. This podcast is for physicians and healthcare providers. If you enjoyed this video, please like, subscribe, and forward to your friends.